Today, we're breaking down the Animal Kingdom chapter with a relentless focus on high-yield NEET exam points. If you've ever felt totally overwhelmed by the sheer number of phyla, examples, and distinctive features in this unit, well, you are in the exact right place. We aren't just going to sit here and read facts. We're decoding the patterns and prioritizing exactly what you need to know to secure your marks. So let's get right into it.
We're kicking things off right away with a massive PYQ alert. You absolutely need to lock this broad classification of kingdom Animalia into your memory. Why? Because this single classification hierarchy alone accounts for nearly half the marks you're going to see from this unit on exam day. Literally half. If you understand how this tree branches out, you can eliminate wrong options in seconds. Notice how it starts by dividing animals based on their level of organization, cellular versus tissue or organ level. From there, it branches into symmetry, mostly asymmetrical, radial, or bilateral. And finally, for those bilateral animals, the ultimate deciding factor is the body cavity or the coelom. So whether an animal is an acoelomate, pseudocoelomate, or a true coelomate instantly narrows down its phylum. Keep this overall structure in mind because it's basically the roadmap for everything we're about to discuss.
Here is exactly how we're going to secure your marks today. We'll start by cracking the classification basis, then dive into the non-chordates, from Porifera to Echinodermata, outline the defining features of chordates, and wrap it all up with a rapid-fire 30-second high-yield recap.
Section 1. Cracking the classification basis.
Okay, let's look at those highly tested coelom types we just mentioned. The presence or absence of a cavity between the body wall and the gut wall is a massive deal in animal classification. When you're looking at a true coelomate, the body cavity is completely lined by mesoderm. You'll see this in annelids, arthropods, all the way up to chordates. Now, consider the pseudocoelomate. Notice how the mesoderm isn't a continuous lining. Instead, it's just kind of present as scattered pouches between the ectoderm and endoderm. When you hear the word poser coelom, I want your brain to immediately scream Aschelminthes. That is a classic high-yield exam association. Finally, there are the acoelomates, where the body cavity is entirely absent, which happens to be the defining characteristic of Platyhelminthes, or flatworms.
Moving on to another really critical basis of classification, segmentation. But you've got to be careful here, because you cannot confuse generic segmentation with true metamerism. This is a vital term. Make sure you mark it in your NCERT. Metamerism isn't just about superficial lines on the outside of a body. It's a phenomenon where the body is externally and internally divided into segments with a serial repetition of at least some organs. The absolute gold standard example of this pattern, the earthworm, which falls under phylum Annelida. When the exam asks you about metameric segmentation, Annelida should be your immediate, zero hesitation reflex.
And that brings us perfectly to our next classification basis, the notochord. So what actually is it? Well, it's a mesodermally derived, rod-like structure formed on the dorsal side during embryonic development in some animals. The presence or absence of this single, specific structure strictly splits our entire animal kingdom right down the middle into chordates and non-chordates. If you don't form this structure at any point in your life cycle, from Porifera all the way up to Echinodermata, you're a non-chordate. It's a highly specific, totally defining anatomical landmark.
Section 2. Non-chordates. From Porifera to Echinodermata.
Let's start with the mostly asymmetrical Porifera, which we commonly know as sponges. Think of examples like Cycon, Euspongia, and Spongilla. These are really primitive multicellular animals, and what you absolutely need to note here is their cellular level of organization. They don't even have true tissues yet. Their entire existence revolves around a central cavity called a spongiocele. Water enters through these tiny minute pores called ostia, travels into the spongiocele, and then exits through the osculum. Also, don't forget the choanocytes, or collar cells, that line this cavity. They are completely unique to Porifera and heavily tested.
Okay, stop everything and listen closely for a second, because this is a classic exam trap that you must avoid. Both Porifera and Echinodermata have systems with the word water in them, and test makers absolutely love to swap the terminology to trick you. Phylum Porifera possesses a water transport or canal system, which is mainly used for food gathering, respiratory exchange, and waste removal. But if you jump ahead to phylum Echinodermata, the spiny-bodied animals like starfish, they possess a water vascular system. The key difference in function? The water vascular system actually helps echinoderms with locomotion, along with capturing food and respiration. So memorize this distinction right now. Transport is Porifera, vascular is Echinodermata.
Moving on to coelenterates, or cnidarians. For this group, their undeniable defining feature is the cnidablast, or cnidocyte. These structures contain stinging capsules called nematodes, which are present on their tentacles and their body. They use these cnidoblasts for anchorage, defense, and of course, capturing prey. Another highly tested point for cnidarians is their two basic body forms. You have the sessile, cylindrical polyp form, like Hydra, and the free-swimming umbrella-shaped medusa form, like Aurelia. Some amazing cnidarians even alternate between these two generations in a process called metagenesis. Pretty cool, right?
Section 3. Chordates, the defining features.
All right, this comparison is an absolute must-memorize, high-yield checklist mapping exactly to NCERT Table 4.1. It breaks down the undeniable differences between the two groups. Now, we already know chordates have a notochord, but consider point number 2. The central nervous system in chordates is dorsal, hollow, and single. Contrast that with non-chordates, where it's ventral, solid, and double. Also, chordates feature a pharynx perforated by gill slits, a post-anal tail, and, this is a big one, a ventral heart. If a non-chordate happens to have a heart, it's situated dorsally. You've got to lock in these structural opposites.
Dig in just a bit deeper, Phylum Chordata is divided into 3 distinct subphyla, and you need to pay very close attention to exactly when and where the notochord is present in each. In Urochordata, like Ascidia, the notochord is present only in the larval tail. It actually disappears later on. In Cephalochordata, like Branchiostoma, the notochord is the ultimate survivor. It extends from head to tail and is persistent throughout their entire life. Finally, in Vertebrata, the notochord is present during the embryonic period, but in adults it gets replaced by a cartilaginous or bony vertebral column.
Section 4. Our 30-second high-yield recap. All right, let's lock this all in. In under 30 seconds, scan these facts. If you see Platyhelminthes, immediately think bilateral, acoelomate, and a flat body. For Aschelminthes, connected strictly to the pseudocoelomate body cavity. When Annelida comes up, your brain should jump to true coelomates with metameric segmentation. For Arthropoda, which is the largest phylum by the way, focus on their chitinous exoskeleton and jointed appendages. And for Mollusca, look for that external calcareous shell. Linking these specific distinctive features to their coelom type is literally the ultimate cheat code for this unit.
I'm going to leave you with this classic NCERT line to truly test your understanding. All vertebrates are chordates, but are all chordates vertebrates? Think about the protochordates we just discussed, the urochordates and cephalochordates. They have a notochord, which makes them true chordates, but they never develop a vertebral column, so they aren't vertebrates. It's an elegant distinction that really sums up the beauty and precision of animal classification. Keep reviewing these distinct characteristics, avoid those common terminology traps, and use this explainer as your ultimate study weapon. Keep up the great work.